Acute inflammation is a complex biological response of the body to harmful stimuli, such as pathogens, tissue injury, or irritants. It is a tightly regulated process involving a cascade of cellular and molecular events aimed at eliminating the injurious agent and promoting tissue repair. The pathophysiology of acute inflammation can be described in several stages. Number 1 is recognition of harmful stimuli. The process begins with the recognition of harmful stimuli by the innate immune system. This recognition can occur through various receptors, including pattern recognition receptors, such as toll-like receptors, which detect specific molecular patterns associated with pathogens or tissue damage. Stage 2 is vasodilation and increased vascular permeability. Upon recognition of the harmful stimuli, the immune system triggers the release of inflammatory mediators such as histamine, prostaglandins, and leukotrienes. These mediators cause vasodilation of local blood vessels and increased vascular permeability, leading to the leakage of plasma proteins and fluid into the surrounding tissue. Stage 3 of acute inflammation is migration of leukocytes. The increased permeability of blood vessels allows leukocytes, particularly neutrophils, to migrate from the bloodstream to the site of injury or infection. This process is facilitated by the expression of adhesion molecules on the surface of endothelial cells lining the blood vessels, which interact with complementary receptors on the surface of leukocytes. Stage 4 is chemotaxis. In response to chemical signals such as chemokines released by damaged tissues, leukocytes migrate along a concentration gradient towards the site of inflammation. This directional movement, known as chemotaxis, helps to concentrate the immune cells at the site of injury or infection. Stage 5 is phagocytosis and clearance of pathogens. Once at the site of inflammation, leukocytes, particularly neutrophils and macrophages, engulf and destroy pathogens through a process called phagocytosis. These immune cells release various antimicrobial substances and enzymes to kill the pathogens and degrade cellular debris. Stage 6 is resolution of inflammation. As the injurious stimuli are neutralized and the tissue damage is repaired, the inflammatory response is resolved. Anti-inflammatory mediators, such as interleukin-10 and transforming growth factor beta, help to dampen the inflammatory response and promote tissue healing. Apoptosis, programmed cell death of neutrophils and macrophages, also plays a crucial role in resolving inflammation. Stage 7 and final stage is tissue repair. Following the resolution of inflammation, tissue repair and regeneration processes are initiated. Fibroblasts proliferate and produce extracellular matrix components, such as collagen, to replace damaged tissue. Angiogenesis, the formation of new blood vessels, helps to restore blood supply to the injured area, facilitating tissue healing. Overall, acute inflammation is a dynamic and highly regulated process that plays a crucial role in the body's defense against infection and injury. However, dysregulation of the inflammatory response can contribute to the pathogenesis of various diseases, including autoimmune disorders, chronic inflammatory conditions, and tissue damage associated with prolonged or excessive inflammation. Thank you for watching Nextmeds. Please like and subscribe for daily videos about health, nutrition, and diseases.